Hello and welcome back to Biology for Beginners. As always, I'm your host, Morgan. This week we're beginning at Chapter 12, which will wrap up Unit 2 for us. Before we really jump into this chapter's topics may seem kind of heavy and hard to digest, and that's because they are. This is one of those chapters in the book that if you were taking an intro-level biology class would for sure be skipped. This chapter is all about DNA technology and genomics, which are basically applied science of what we've talked about in the previous chapters of this unit. So without further ado, let's talk about gene cloning and editing. As always, right now you should see our terms and definition slides popping up around me. And if you would like, feel free to pause and grab the vocabulary we will be using today. However, as always, I have a reminder for you that if you would like a PDF copy of my slides for absolutely free, please head on over to my Patreon and you can find it there, as well as any other study material that I have produced to go along any of my Bio for Beginner videos. However, if you are a patron, you will get the Biology for Beginner material at least a day in advance, sometimes more. Let's get into this. What is biotechnology exactly? Well, biotechnology is pretty much the manipulation of living things or parts of living things to make something useful. Historically, biotechnology has basically been limited to selective breeding to get things like the domesticated farm animals, like the dairy cows, specific crop plants like modern corn we see today. The wild ancestors look very different, and also breeding dogs to make specific breeds to carry out particular tasks, such as collies for herding or hunting dogs and things like that. However, today when you hear the word biotechnology, you're probably talking about a more specific part of biotechnology known as DNA technology. And DNA technology just means you're actually manipulating genetic material in the lab. And again, it's usually to produce something useful. So a little bit more about DNA technology here. So DNA technology is specifically studying and manipulating genetic material in the laboratory setting. So an example of that is the extraction of genes from one organism and transferring them to another. So like the glowfish you see here in the photo on the slide, this is an example where you took a fish, you added some sea jelly DNA, and then you get the glowfish, these little fish that are bright colors that glow lovely under black lights. And DNA technology kind of kicked off in the 1970s. And that's when making recombinant DNA became possible. And recombinant DNA is DNA that's formed by combining the DNA of two different sources. So DNA usually from two separate species, like the glowfish, which you can walk into any Petco or PetSmart now and pick some up from their aquarium department if you would like to have a fun little aquarium for your kids or for yourself. I'm not going to judge. And recombinant DNA is most widely going to be used for genetic engineering. And genetic engineering is just the direct gene manipulation. And again, this is all DNA technology. So you're taking genetic material in the lab and then manipulating it in some way. And if you're combining DNA from two sources, you're making recombinant DNA. And if you're just kind of manipulating genes, that's just genetic engineering. Let's talk a little bit more about gene manipulation. Now, say you're a biologist and you want to manipulate genes. Chances are you're probably going to use some form of bacterial plasmid. And recall that a plasmid is just a small section of DNA that can replicate separately from the main bacterial chromosome. If you want a little bit more on that, please go back to our chapter 10 videos where we talk about that replication and all that fun stuff in a little more depth, there is a little bit of a section specifically about plasmids. Plasmids are the perfect tool for something known as DNA cloning. And DNA cloning is just when you have a section of DNA and you want to make identical copies of it or clones of it. So you start with one copy of that DNA and make more. And the reason plasmids are a perfect tool for this is because they only carry a few genes. They can be passed on from one generation to the next. And remember, they replicate independent of that bacterial chromosome. So the gene you're trying to copy is not going to get mixed up with all of those other genes. And they can be easily transferred into bacteria. That is a big deal. And we'll talk a little bit more about that in the next couple of slides. And because they're so small, they can be easily manipulated and carry almost any gene you want to put on them. So all of these reasons are the things that make plasmids perfect tools for DNA cloning and the perfect tools for gene manipulation. So let's talk about the steps a little more so you can kind of visualize what goes on. And again, this is more of a general idea of what goes on. If you actually go into the field where you're going to be manipulating genes and all that, it may be slightly different depending on what methods, but this is the dominant method. And it may be a different bacteria depending on what organisms you're working with. So step one, you're going to isolate a plasmid. So that plasmid that you want to use has to be removed from the bacterium. And usually it's going to be an E. coli bacteria. And that's just because, like we've mentioned before, E. coli, or what is known as a model organism, they're easy to propagate in the lab and they're pretty plentiful. So you isolate that plasmid. And the plasmid is going to act as something that is known as a vector. And basically, the vector is just the thing that's going to carry the gene of interest for you. And we have the full definition in those terms and definition slides if you would like to know the more technical definition. But the big deal here is your vector carries the thing that you want. And so then you're going to move on to step two, which these first four steps kind of occur at the same time. So keep that in mind. The DNA that's going to be from another source that's going to contain that gene of interest for you needs to be isolated. So the DNA is going to be kind of pulled out of the cell, and you're going to pinpoint that gene of interest and move on to steps 3 and 4, where you use a specific enzyme to cut both the plasmid and the DNA. And the important part here is that you use the exact same enzyme for step 3 and 4. And that will ensure that the DNA from the plasmid and the DNA and the gene of interest complement each other, so that in step 5, once that cut DNA from the plasmid and the gene of interest are mixed together, you can move on to step 6, where DNA ligase, which is just an enzyme that occurs naturally in the body that basically pastes parts of DNA together, can join those two pieces of DNA, and you can end up with your recombinant plasmid. And again, recombinant just means that you have DNA from two sources. So you've recombined, mixed things up, and resulted in that recombinant plasmid containing your gene of interest. And then in step 7, you're going to mix the recombinant plasmid with bacteria. And again, it's usually going to be E. coli. And then, since bacteria can take up new genetics through transformation, if you want more information on that, please see previous videos where we talk about genetics and specifically those single-celled organisms. So transformation is basically where a single-cell organism takes up new genetics from the environment. And because plasmids are little pieces of DNA and genetic material, bacteria will usually take them up pretty readily, and that's what happens in step 7. And then step 8, the recombinant bacterium is just allowed to reproduce. And remember, the main way bacteria reproduces asexually through binary fission. So the chromosome, the plasmid are going to replicate, and then it's going to split in 2. Those 2 are going to split in 2, so on and so forth. And they're all going to be clones of one another. So they're all going to be identically similar, or genetically identical. And because they're genetically identical, that means you've just made a bunch of copies of your gene of interest as well. And then in step 9, you have 2 possible pathways depending on what you wanted to do. Pathway A, you were just trying to produce a lot of a specific gene to get a lot of a specific protein made. And so then once you have enough made, you begin to harvest and use those proteins. And this happens a lot when you're harvesting the proteins for insulin for people with diabetes, and also proteins that are used to treat blood clots. Or the second pathway, where these genes are actually inserted into other organisms, which we will talk about more in another video. But an example of that is inserting a gene for pest resistance into plants like corn. Let's talk a little bit more about cutting up and pasting the DNA together, since we kind of glossed over it in the last few slides when we were talking about the steps of gene manipulation. So the first part is the cutting up of the DNA. So what we use are these bacterial enzymes. So they're enzymes that are already present in bacteria, which is great because that means we don't have to make them. And these enzymes are called restriction enzymes. And the reason restriction enzymes exist is because bacteria use them to cut up foreign DNA and protect themselves from things like viruses and other bacterial parasites. And restriction enzymes recognize these really short sequences of nucleotides, and these little sequences are known as restriction sites. And these are the sites where the enzyme can attach to the DNA and then cut it up. And it's as simple as that. So when it's cutting up the plasmid, it just kind of cuts a nice chunk where the gene of interest can then be inserted, and then it'll cut around that gene of interest if everything is done correctly, because it will recognize restriction sites on the DNA that contains that gene of interest. But once all of that is cut up, it needs to be pasted together. So let's talk about pasting DNA together. Now, the 2 pieces of DNA are going to complement each other because they've been cut up with the same enzyme. As you can see here in this diagram, there are these kind of sticky ends that will recognize each other when everything's mixed together and form kind of weak hydrogen bonds. And once they are kind of loosely pasted together, DNA ligase, which again is just another naturally occurring enzyme that is going to be hanging around when DNA is replicated normally anyway, because its entire job is to turn those temporary bonds into permanent bonds. And then you result in this recombinant DNA, and you're pretty much just using things that occur naturally anyway. You're just manipulating it in the laboratory setting for a specific goal. So how do you pinpoint a gene of interest? Well, one thing you can do is you can utilize something known as a nucleic acid probe. And what nucleic acid probes are they're these little radioactive segments of single-stranded DNA that complement your gene of interest. And you can take these probes and mix them in a solution with the DNA, and it's going to end up forming hydrogen bonds with the target gene because, again, it complements the target gene. And the good thing about these nucleic acid probes is you can apply them to multiple DNA molecules at once, and you can see where this gene is on the different molecules of DNA. And there's multiple practical applications for these probes. It's not just for finding a gene to be cut out of a piece of DNA, stuck into a plasmid, and then used for something else. But we're not really going to get into the more technical things. The big idea here is that there are these little radioactive segments of DNA that hydrogen bond with a specific gene because it complements it, and it allows a researcher to then visualize it because they are radioactive. They will glow under the correct situation. So one other thing we need to talk about now is using reverse transcriptase in gene manipulation and all this fun stuff. So reverse transcriptase is another enzyme that comes from what we know as retroviruses. Remember a few videos back, we talked about viruses and we talked about how retroviruses have an enzyme that allows them to reverse the flow of genetic information. So normally it goes DNA to RNA to protein. However, a retrovirus has reverse transcriptase, which is an enzyme that allows RNA to then be moved into DNA. So in the lab, they've been able to utilize reverse transcriptase to take mRNA and use that to then synthesize DNA and result in what is known as complementary DNA. So basically, you've got the DNA in the nucleus, you've got the mRNAs you want in the cytoplasm, you take those, put them in the test tube, and then in the test tube, there's the addition of reverse transcriptase, and this reverse transcriptase synthesizes that new strand of DNA. The RNA will then eventually break down, and then a second DNA strand will be synthesized. It's a little more in depth than what the book goes into and what we're going to go into here. But again, this entire chapter is one that would probably be omitted from a normal intro-level biology class anyway. But the point of this series is just to march through the book. So we're at least giving you a little taste of some of this. So I know that I've kind of glossed over the last couple of slides, but that's the reason. And we're going to gloss over this last slide here as well, because this is more of an experimental gene manipulation that's going on, and it's just kind of a fascinating thing. So all of the previous methods of gene manipulation that we've looked at have been done in vitro, which just means outside of living cells. You do all of the manipulation basically in a test tube and then stick it back into a living cell. Well, with this new kind of exploratory thing that they're looking at, the CRISPR-Cas9 system allows editing within living cells. So basically they use a different protein, so the Cas9 protein that cuts DNA, and then the segments of DNA are joined. We're not going to get more in depth in it than that. You wouldn't need to know about it in an intro-level biology class either, but it is a fascinating little bit of technology that's being worked on. It's just something that intro-level bio students aren't going to be expected to really know about. It might be mentioned tangentially in a class, as most of the content of this video would be mentioned probably tangentially in a class. Honestly, the main steps of gene manipulation are likely to be the only things from this section you would be expected to know anyway. But if you do want to know more about it, I do encourage you to look into it. It's fascinating. It's not something I'm super familiar with because my specialty isn't in this more molecular genetic biotechnology side of bio, but it is fascinating. And again, if this section was kind of hard to digest, don't feel bad. It is a very heavy, hard to digest section, and the next couple of videos following this one will probably feel similar as well. But hang in there. I hope you have a wonderful rest of your day, afternoon, evening, what have you. Don't forget to like, comment, and subscribe, and I will see you all in the next one. Thank you again for watching this video. Don't forget to like, comment, subscribe, and ring that bell if you would like to see more. And if you'd like to follow me on any of my other social medias, the links are down in the description below. Don't forget to check out thereptilegoth.com for all of my articles and blog posts. If you found any value in this video and you would like to help support the channel, please check out my Patreon page. That link is also in the description down below. And a special thanks goes out to my Diamond Dragon patron, Diane V. What you're doing is really helping me fund a dream here. I will see you guys all in the next one.